going to introduce Dr. Matthew State. Matthew State, MD, Ph.D., received his undergraduate and medical degrees from Stanford and completed his residency in psychiatry and fellowship in child psychiatry at UCLA. He got his Ph.D. in genetics from Yale and joined the faculty in 2001. He is now the Donald J. Cohen Associate Professor of Child Psychiatry, Psychiatry and Genetics, the Vice Chairman for Research in the Department of Psychiatry, and he co-directs the Yale program on neurogenetics. Dr. State's lab has been a leader in highlighting the contribution of rare genetic variants to childhood neuropsychiatric disorders, including Tourette's, autism, and mental retardation. In 2004, his lab was the first to show that the contactin family of brain expressed molecules was involved in both intellectual and social disability. In 2005, they identified the first genetic mutation associated with Tourette's disorder. And recently, with his collaborator and co-director of the Yale program on neurogenetics, Murat Gonel, he published one of the first studies to use next-generation whole exome sequencing to discover genes involved in developmental brain disorders. The latter two findings were cited by Science magazine as among the top 10 scientific breakthroughs the year they were published. Dr. State is currently leading a large multi-site genome-wide study of autism spectrum disorders funded by the Simons Foundation and is principal investigator on multiple NIH grants focusing on both autism and Tourette syndrome. Dr. State.
Good afternoon. I hope you like the introduction. I have to tell Mom she wrote it, and I thought it went very well. What did you think? All right. So I'm going to talk today about Tourette syndrome. So this morning I talked about autism. I don't know. I'm going to see if there are any familiar. No, probably not. As you just heard, we focus on gene discovery in childhood psychiatric disorders predominantly. And we've been working for now more than a decade, both on Tourette and autism, and then intellectual disability and structural brain disorders. This morning, I talked about a large-scale study in 1,000 families doing work in autism. And today, this afternoon, I'm going to switch and talk about sort of the other end of the spectrum. Really just one family and a rare genetic finding. And I'm doing that in part, it would have been nice but way too long to put them together, because one of the questions I think that comes up is, what do you do with genetic findings, particularly rare ones? So you find them and then, how does that help? And then we talk about, this morning it was, if we can find molecular mechanisms underpinning autism spectrum disorders or schizophrenia, that may open up novel avenues for treatment. And this afternoon, I'm going to try to convince you that that really is a possibility. But in this case, I'm going to talk about how that is playing out now with regard to Tourette syndrome. And I just want to make sure I don't see the IT people that am I audible? They left, so I'm good. Okay, so I'm going to start where we start, which is in the clinic. Actually, this is HBO's clinic. But I wanted to show you, because I figured, since I guess many of you are not child psychiatrists, and we mainly see kids with Tourette, so I thought it might be worthwhile to show you some of the clinical phenomena.
Is a certain brain defect. It's a neurological problem that I was born with that causes you to tick. Ticks control our bodies. Ticking, like coughing, twitching, like snapping all the time. Do a lot of things people wouldn't like. My mom calls me the mouse because I make a little squeaking noise. Shaking my head. Now that I said it, I have to do it. I'll go like this, flapping my arms, and everybody thought I was trying to fly. I'm like, no, you can't help it.
Okay. So actually, I don't know whether I can pause it. It's just going to run this again. I'm going to move to a second video, but the first one, we just wanted to show you a number of kids who are showing sort of classic signs of Tourette syndrome, which is simply the combination of vocal and motor tics. So you saw lots of examples. But they are semi-voluntary, sort of ultimately irrepressible. If I asked you to not blink for the remainder of the talk, you would do okay for a few minutes. You would be able to not blink, but ultimately the blink would be irrepressible. And when we talk with kids about the performance of their tics, it has that quality. So that if a parent says, please stop that, particularly that happens before parents understand why the kid is making squeaking noises in the back of the car, for a short period of time the child will be able to suppress the tic, but ultimately it is involuntary. But so we think of it as a semi-voluntary movement disorder. It has a waxing and waning clinical course. But I want to now show you a second video that goes a bit more in depth into kind of the internal world of kids with Tourette, and this is fairly representative of what we hear when we start doing clinical interviews. That's not what we hear. This is what we hear.
Noah, 4 times. 1, 2, 3, 4.
Yeah.
4 times instead of 4. Instead of 4.
Yeah.
And before I go to bed, I have to go to bed. And I lay down. I have to look at my clock. Picture the time in my head. So I sort of not memorize the time, but make sure I know it. And then before I go to bed, when I'm in bed, I have to blink 4 times, 4 sets of 4, 4 sets of 4, 4 times.
And are you having any vocal tics now?
Yeah, I still go and little words like that.
Is there a feeling that goes along with those, or they just come?
They just, well, sometimes I have to get them just at the right pitch. At the exact pitch that I want. It's either, and sometimes when I talk, I have to do it like an opera kind of voice. Like a loud, high-pitched tone. And then now I say, instead of saying the F word, I say fuck instead. That's a little better.
And is there a particular time when you have to do that?
Well, I don't swear anymore. I just, whenever I get that little feeling inside that I have to do it every morning. But I think it's really terrible to see people making fun of other people for something they cannot even help. Any disease I'm talking about, anything. They can't help that. And I can't help this that I have, and I don't want anyone making fun of me. So if you see someone who's doing not odd, but unusual things, don't make fun of them, because you don't know. Some kids have been gifted and don't have anything, and some kids have just had something because genes or their family.
So of course Chelsea gave us permission to watch the video, and the one deal was that I told her that I would always leave in that last part, because she was really determined that when I teach about Tourette syndrome that people would hear her talk about that. Really the biggest disability for her, particularly through middle school and the early part of high school, was the social ostracism that came with making operatic sounds as she would walk down the hallway in school. It was somewhat brutal for her. Things are going very well. She's doing beautifully now. But in any case, and like with most things, if you listen carefully to patients, you'll hear a tremendous amount. So she talked about sort of the internal rising sense of tension that she responds to by performing these actions. She has a feel just right phenomenon, which we find in a significant proportion, in some studies 90-95% of folks who have TS, and this very kind of similar phenomenology to obsessive compulsive disorder, right? There's it happens and I have to do something in order to get rid of it, and really in the clinic we see a tremendous overlap between pure obsessive compulsive disorder and Tourette syndrome, and sometimes it's quite difficult to separate out complex motor tics which might involve multiple motor movements and vocalizations. To separate that from OCD, and the way that we do it is we say, well, if it's in response to a formed thought about anxiety, then it's OCD, and if it's in response to just this kind of inchoate feeling, a rising sense of tension, then that's a complex motor tic. I think that whether or not that has any validity, we don't know. That's what we do in the clinic. But the reason I bring it up is just that from a phenomenological standpoint, and also, as I'm going to talk about in a minute, from a genetic and neurobiological standpoint, the kind of overlap between obsessive-compulsive symptoms of obsessive-compulsive disorder and Tourette syndrome is quite high. We see more than half of the kids that show up in our clinic for treatment for TS will have comorbid obsessive-compulsive disorder, although I don't really know what it means to be comorbid, whether or not these are two things that are happening simultaneously, or there are studies that suggest that they have a shared genetic origin and certainly a shared neurobiological origin, so it's tough to kind of sort that out. What I want to do briefly is I'm going to give you the sort of briefest overview about the neurobiology of TS, just to give a little bit of a sense and focus in on sort of one aspect of that, because it'll become important later in the talk. So just a couple slides on neurobiology, a couple slides, sadly only really one slide on the genetics of Tourette, because we really know very little about it, and then I'm going to tell you about some of the recent data in the lab. So this is my, I hesitate to say artist's rendition. This is my rendition of cortical striatal circuits. It's sort of where everyone goes when they think about neuropsychiatric disorders, and essentially what I'm trying to do is to represent the can we see it? Multiple layers of the cortex, and then with excitatory neurons that project down into the striatum, and then there's a loop back through the thalamus, and then back to the cortex. So these are cortical striatal thalamocortical circuitry. And for a whole variety of reasons, we think that somewhere in the circuit is where obsessive-compulsive disorder and Tourette syndrome originates. Because it's a circuit, you can imagine that you could mess it up in a whole variety of different spots and end up with kind of a similar outcome. So even though we have a sense from neuroimaging studies, animal studies, some human lesioning studies, that these deep nuclei, the basal ganglia, I don't know whether you can see where I am, are key, it really is kind of at a 30,000-foot level. We know that when we do neuroimaging studies, and we look at tic suppression studies, or we do studies of OCD, that we see changes in the bold response in these areas. But whether or not that is driving or compensating for the genesis of these disorders, we really don't know. I'm not sure that I need to show you a whole lot more than that, but just to suggest that when we think about Tourette syndrome, we think about the striatum, and as a general proposition, what we know about this really is kind of at that level, that we don't know about the cellular molecular mechanisms that might drive TS. We don't understand, as a general proposition, what genes are involved. What we have an idea about is that if you're thinking about Tourette, that this circuitry is likely to be the place to look. Again, in part by what we found in neuroimaging studies of Tourette, but in part by analogy also to other movement disorders like Parkinson's disease and others that also live in the striatum. So the one other thing that I want to point out, again, this is just a setup to the story that we're going to discuss, is that there's been a tremendous interest for a long time in the role of dopamine and dopaminergic neurotransmission in Tourette syndrome. And really the reason that's there is not because of some major insight, at the molecular and cellular level. It really comes from pharmacological observations, or observations of pharmacological phenomena. So on the one hand, we know that we can effectively decrease tics by blocking dopamine. So atypical and typical antipsychotics will reliably decrease tics in the short run. Now, tics have a tendency to wax and wane, and so they almost always come back to some degree. But if there's a bad tic and you want to suppress it, the largest effect sizes are for dopamine blockers. And conversely, we also know that a substantial subset of kids who are treated with stimulants that are dopamine agonists will develop tics. Some of them will develop tics, and if they have a previous family history of Tourette syndrome, or even if they don't, sometimes the tics persist, and that's a relatively small proportion of people. But there is a substantial minority of kids who have no family history and who do not have Tourette syndrome, who you simply give stimulants to, and they will have these abnormal movements. And whether any of you treat ADHD, but it's not infrequent to run into trouble, and it's quite dose related. If you stop the stimulant, they'll stop ticking. If you give the stimulant back, they will tick some more. So now, So dopamine seems to be important, but again, when we've done studies to try to use that as a hypothesis to understand the underlying mechanisms, we haven't gotten that far in Tourette syndrome. So there have been neuroimaging studies about the role of dopamine, and there really has been no smoking gun. There are no reliable, reproducible results with regard to dopamine signatures, say with PET imaging, and there have been genetic studies looking at common genetic variations in things like the dopamine transporter, et cetera, and there have been no reproducible studies that are either showing that those genes or variations in those genes are contributing to Tourette syndrome. So, now, it probably should be in a separate slide, but while we know that dopamine blockers have the largest effect from the standpoint of pharmacological treatments, we don't have great treatment options. So the issue with dopamine blockers is they work in the short run, but then we have difficulty both with increasing tics with withdrawal. So there seems to be hypersensitivity, which corresponds with removal of dopamine blockers, particularly from high doses. We all know, I would imagine it's mostly psychiatric audience, about difficulties both with typical and I think what's been more in the news lately is that atypical antipsychotics are far from benign, and particularly when you're giving them to kids, that relegating a child to long-term treatment with an atypical, and then the problems with metabolic or potential metabolic syndrome, diabetes, weight gain, etc. It's not a wonderful treatment option. We also have this problem that tics wax and wane and generally get better in adulthood. It can be bad enough that you really want to treat in childhood, but in the back of your mind, you know that if you hang out long enough, that there's a good chance that the child's going to get better, and that makes it even more difficult at times to think about, again, relegating someone even with a bad tic to a prolonged course of treatment with antipsychotics. Another option are alpha agonists. We use clonidine and guanfacine as treatment options, and they're much more benign from a side effect standpoint, but they have much smaller effect sizes. And many clinicians will tell you that the effect size is 0. Studies say that it's better than that, particularly over the long run, but they're certainly not great medications if you have a bad tic that you want to treat or a series of bad tics. Really, actually, the best therapies right now in terms of the balance of effect sizes and side effects is behavioral treatment. So there are particularly behavioral treatments that are quite effective. The problem is they're very hard to get at. It's essentially a modification of habit reversal therapy, and they can work very well, particularly for isolated, very bad tics. But there are only a handful. As you know, in the child world, it's very hard if you're not near a tertiary care center to be able to get someone who has Tourette in for a trial of CBIT, which is the behavioral therapy. So it would be good if we could use an understanding of molecular mechanisms to come up with a better way, a more direct way, a more biologically driven way of attacking Tourette syndrome. And there has not been a tremendous amount of activity, as you might imagine, in the pharmaceutical industry around Tourette, in part because even though it's relatively common, you'll see on this slide it's somewhere in the neighborhood of 0.5 to 1% of the population. There haven't been a lot of clues. There are not a lot of leads to pursue. So it would be good if we could think about having another option. I've already told you about the chronic motor vocal tics associated somatosensory phenomena. There's a sense of just right, almost an aura that surrounds sometimes the performance of a tic. The natural history is onset at school age with a waxing and waning course. And as I said, typically improvement in adulthood, about 70% of patients we see in clinic. But sometimes it can be very hard to wait and see whether or not that's going to happen. And then we have very high rates of comorbid disorders. Now, from a genetic standpoint, there was early on, Tourette was thought to be was going to be the next thing after Huntington's disease to fall to contemporary genetic methods. The idea was it was a single gene disorder that was dominantly inherited. This came out of some studies of very large pedigrees that were found in Utah and in Oregon with hundreds of people, and the pattern looked like simple inheritance: one gene, one disorder. There's a much higher rate of seeing monozygotic twins sharing Tourette or Tourette and OCD than dizygotic twins, suggesting that there is a strong genetic component. The bottom line here is going to jump ahead is simply that while the pedigrees looked really great, the bottom line was as genetic technology advanced and it became essentially impossible to lose something that was hidden in the genome if it had this big effect, no single gene was found. And subsequently people went back and realized why they were kind of misled by these pedigrees that looked like this was dominant inheritance. So it turns out, for reasons that aren't entirely clear, that spouses in families, particularly where there's more than one offspring that have TS, are very likely to both either have the disorder, either have Tourette syndrome or OCD on both sides of the family, or have strong family histories on both sides of the family. So there's a very high rate of what we call bilineal inheritance, and about 40% of families, if you see them in clinic and there more than one kid is affected, you will find that both sides of the pedigree have TS and or OCD. And for geneticists, that can be really problematic. If you're trying to trace inheritance, you really want it to be coming from one side of the family so you can trace it. When it starts coming from two sides of the family, particularly multiple generations, it's essentially impossible to use standard methods to sort of sort that out unless it's a recessive disorder. But something like this, what happened is that the families looked like about 50% of offspring were having it, and they were really focused on fathers transmitting it from these big Utah pedigrees, and then when they were unable to find the gene, they went back and realized that they had not done a comprehensive job of asking about the people marrying into the families, and it turned out in these large pedigrees that there was a huge rate of bilineal inheritance. So what looked simple lit up like a Christmas tree and did not any longer look like dominant inheritance. All right, so we've got, this is really the story of psychiatric genetics. We thought it might be simple. It wasn't simple. I could substitute schizophrenia, autism, bipolar disorder, anything else and tell you what we think is this is a multigenic disorder, that there will be multiple genetic variations that can contribute to this. And we don't know a lot about what the particular mix of variations might be, how many genes might be contributing, and whether or not the variations in the genome that might be responsible are common or rare in the population. Now, what we do know is that so far studies have not found a lot. So the very large pedigrees initially turned out to be misled by this bilineal inheritance probably, and by a multigenic underpinning. Then there was kind of this shift, again, which is similar to everything else in psychiatric genetics for a while. They went to different kind of linkage methods that were supposed to be able to be more robust to these kinds of problems. The studies were very small. They found nothing. Then we went to picking our favorite gene and asking whether or not that was responsible, called the candidate gene association study, and we found not a whole lot there. And now we're in the age of what's called genome-wide association. And genome-wide association is you can stop trying to pick your favorite gene. You take a look at every gene in the genome, essentially, and then you ask the question, is there any variation in a gene around the genome that is more common in your cases than in your controls? And it's been very powerful in many areas of medicine. Right now in Tourette, so far, there's one genome-wide association study that's currently underway. It's at a size where in other disorders we started seeing, not in psychiatry, but say in aneurysm or diabetes or something like that, where we started to see some evidence for these, for common genetic polymorphisms contributing, and so far we're not seeing it in Tourette. So that might sound like we're really in trouble here. But one of the things I want to point out, this is from a recent review. I'm not sure whether it's even online yet, but there is that I wrote it for Current Opinion in Genetics and Development, and I have this kind of pet peeve because everyone's like, well, Tourette, it's been such a mess. It's like it's worse than autism. It's worse than schizophrenia in terms of not being able to find anything. But so I thought, well, it would be interesting to go back and just count the number of genetic studies that have been done on Tourette syndrome over the last decade and take a look at how that compares with disorders where we're making progress. So this is a really kind of rough, this is the number of annual publications. I started in 1985 and then just, PubMed search, Tourette genetics, autism genetics, schizophrenia genetics, tallied them up, tried to take out the reviews. So it's a pretty rough estimate, but my guess is that the scale is about right, the orders of magnitude are right. So autism is in red, and you can see that there's been a steady increase starting from like less than 185 in like 2009. We're up to over 300 publications a year, and that's coincided with a lot of advances in autism genetics. It's a very hot area with a lot of good stuff coming out. Schizophrenia schizophrenia, as you can see, is like monstrous. And again, in the last 2 years, both for common and rare variants, people are starting to make real progress. And look at where Tourette syndrome is. Right? This is a really sad slide. So not only can I tell you not a lot about the genetics of Tourette syndrome, but what I can tell you is that there have been no more than 20 publications a year in the genetics of Tourette syndrome over a decade. So all that suggests is that there might be actually a fair amount of traction here that it just has not yet sort of come to the fore. That it's a hard problem, but it may not be an impossible problem, and it would be not a good idea to just simply judge by the rate of progress so far and say that we can't make any headway. And actually what I'm going to try to convince you of is I think we are making a little bit of headway, and even though we're down there at 20 publications a year. So before I do that, I'm going to take just 1 minute to talk to you about the approach that we're taking in the lab and how it contrasts with some of the other popular approaches in psychiatric genetics. So this is a slide that shows a relationship between how common a genetic variation is in the general population and what its size of effect is going to be if it's related to a clinical phenomenon. And so what we know is that there's a general proposition in genetics that very rare genes that are or genetic variations that are related to disease often have very large effect sizes. Right? So these are like, over here are Mendelian disorders where you have one gene, has very high penetrance, is essentially a one-to-one relationship between having a genetic variation in that gene and having a disorder. So you can think like Huntington's disease or sickle cell or something like that all the way over here. And this first graph is sort of where we thought psychiatric genetics was, or genetics in general was, up until about 4 or 5 years ago. And so everyone agreed that variations that were common in the population would have smaller effect sizes. And there was a notion that since we couldn't find these guys writ large in things like schizophrenia, autism, or Tourette syndrome, that this is where all the action was, that we weren't finding genes over here because we were looking at the wrong end. So there was about a 10 or 15 year period where pretty much all of psychiatric genetics was focused entirely on looking at common genetic variation and asking about its relationship to these common disorders. And as a general proposition, studies were powered to find small but not infinitesimally small effect sizes. So we figured a couple hundred people probably should be enough because we think the effect size may be 3 to 4-fold increase in risk, because over here you've got 20, 30, 50, 100-fold increase in risk, right? So this was before, and when this model obtained, we literally were finding essentially nothing. So there's this whole 10 years of candidate gene studies, and it's not just psychiatry, across all of medicine was like, okay, it's a serotonin transporter is this, or it's something else is that, and no one could replicate it, and there were only a handful of things. So you look at meta-analyses in the early 2000 or reviews, systematic reviews, and there were like 700 candidate gene studies reporting positive results. 300 of them they tried to replicate the results, and then there were 7 in 2002 out of the 700 that had anything like reproducible findings. So there was a lot of effort put into looking for common variants one gene at a time, and not a lot of progress. Now, we then switched to looking genome-wide, and there has been much more progress. There are hundreds of regions of the genome that have been identified in common disorders outside of psychiatry. But one of the major points here is that now when we have some empirical evidence about what the effect sizes are for these common variants, they are really, really small. So now you will be laughed out of a study section if you're not powering a study to find a 10% or 15% increase in risk. Not a 4-fold increase in risk, a 10 to 15% increase in risk. And moreover, what's happening is that when you start adding up all the common variants that have been found in common disease, you end up explaining less than 10% of the overall genetic variants. And this is just by way of suggesting that there has to be something else. Right, that we're up to now 50 to 100,000 people in these studies. We're really well powered. We're getting to kind of an asymptote on this where they're just either there are going to be hundreds and hundreds of loci that essentially are carrying infinitesimally small risk, which is, I think, unlikely, or the genetic architecture of these disorders is not all about common variants, and it may not be about Mendelian disorders, right? We know that Tourette syndrome isn't Mendelian, but we may actually have some traction by looking in various sorts of contexts for rare variations that are having relatively large effects, but are not going to be common in the population. And that really has been driving our lab for like the last 10 years, both in autism and in Tourette syndrome. So we're, as my mom said in my introduction, we've been very interested in rare or as she wrote, not said, that we're interested in rare variants. And the idea behind this is that, so it's a pretty minimalist idea. The idea is that within this kind of a gamish of whatever is leading to these complex heterogeneous psychiatric disorders, some small portion is accounted for by genes of that have large effect, mutations that have large effect. Because the strategy is really about using genetics as a means to an end. So our lab is not interested, well actually we would be interested if we thought we could do it, we have no hope that we are going to explain the entire genetic architecture of Tourette syndrome. In fact, if we can explain the genetic architecture of one family and really nail it in Tourette, we'll be happy, because the idea is to use genetics as an entree into biology so that we can get up and not say we know nothing about the molecular and cellular mechanisms of the complex psychiatric disorder. And then the question will be, if we can do that, whether or not this rare mutation that we might find, it's not the mutation itself that might generalize, but it might point to a biological process that is generalizable. And so that's this diagram. The idea is that we find these rare outlier families, and we do this either by finding individual families that still look like they have Mendelian inheritance, and we can do that either by looking for large multiply affected dominant pedigrees, or we go to the Middle East and we try to find consanguineous families, inbred families that look like they have a form of the disorder that is Mendelian. Meaning that the genes have very large effect and essentially have reliable transmission from generation to generation. Then we map the gene using very old school methods, like this is going back to the earliest methods in gene mapping, but now supercharged by new technology. And then once we find the gene, we'd really like to make sure that it's related to the human disease, which is easier said than done. But the idea is that this sets off a cycle of scientific pursuit that goes from the identification of a rare gene to the elaboration of the expression and function of that gene, to the development of animal models to be able to manipulate it in its sort of in a natural context to see what proteins it may be involved with, what kinds of basic biological processes might it subserve, and then we're using that ultimately to clarify the mechanisms of the mutation. And then hopefully what that's going to do is lead us to some opportunity for novel insight into how to treat, right? So this is sort of the ideal. And it's been, in 10 years, we've had some success getting to here, but and actually we've had some success probably getting to here, but getting to here is really tough. It can take quite a long period of time. But now I'm going to tell you about an example that's sort of unfolding right now, which I think may actually be sort of a perfect storm that has moved us around the circle in a way that's faster than we anticipated that it might be possible to do that. And it really starts with this pretty unusual family, actually very unusual family. And for the geneticists in the audience, I know you're looking at it going, that's impossible, but I can talk to you about it later. For the rest of you, the major point is that there is an affected father and 8 living affected kids. You can also see that there are multiple miscarriages. Again, because of the short time, I'll reserve that maybe for questions. But the bottom line is that at first blush, if you count up the total number of offspring, the number of affected people, and you do a careful family history to find out whether or not there's any Tourette syndrome on this side of the family, which we did. There's no Tourette coming from this side, so it's not a bilineal pedigree as far as we can tell. And we look for Tourette syndrome, autism, learning disabilities, and obsessive compulsive disorder on this side of the family. And at least for the few generations where we got a good history, there was no evidence that something was coming from that side. And we have the dad who is clearly affected both with Tourette and obsessive compulsive disorder. And then all 8 kids had clearly diagnosable. They were initially diagnosed by neurologists. We brought them to Yale, had them diagnosed in a Tourette clinic, and it really wasn't that hard to tell. They have vocal and motor tics. They'd been persistent. Many of them were on alpha agonist medications. And 2 of the kids also had obsessive compulsive disorder, and 1 also had Asperger's syndrome. Okay? So now, in this family, this is exactly what we look for. This is not a typical Tourette family. But it begins to give you an idea that you might be able to track down what portion of the genome is in the father and all 8 kids and is not present in the mother. Okay? And so essentially that was our genetic study. Find the region of the genome that's present in the father, all 8 affected kids, and not in the mother. Given this size family, we anticipated that there might be anywhere between 1 and 4 of these regions if we were barking up the right tree. And we thought, that could be problematic because you don't know which one of the 4 to choose. But we got lucky. There was only a single region of the genome that was shared by the father and all 8 kids. Okay? So, and these are the genes in that region. And now, sadly, this was in the olden days, about 18 months ago, when you had the only way to look at this was to go gene by gene. So you go gene by gene. Actually, I have to go back and reintroduce her. She went gene by gene. This is Gulhan Erkan Centicek. She's a postdoc in the lab. And she went gene by gene looking to see whether she could find something that looked like it might explain what's going on in this family. The reason I say the olden days is because now sequencing has moved so quickly that this is literally like a week-long experiment. You take the DNA, stick it into a machine, and we would sequence all the genes simultaneously. The other point, though, that I want to make about, it's not just, in the days of the giants we had to do this one by one, but we actually, tried to be smart about it. And we said, okay, so we got 54 genes in the region. We're going to rank them based on what looks brainy, and then we're going to sequence the brainy ones first, and we'll leave the other ones till later. And so, like SEMA6D, SEMA5A has been found in autism. So we're like, okay, SEMA6D, let's start there. And we sequenced all these genes, and we found nothing until we got to gene 51. Okay, so 51 out of 54, because we thought that's a really bad gene. We don't want that gene. And then we got to that gene, and it was the spot that we found a mutation that was clearly altering protein structure. So we went ahead and completed all 54 so we could say, as I am now, we looked in all 54 genes in this interval, and no other gene had a change in it that resulted in a change in protein structure. And the change in protein structure was what's called a stop codon. So it's a nonsense mutation that lops off half of the protein. And the protein, the gene that makes the protein is called L-histidine decarboxylase. It is the rate-limiting enzyme in the synthesis of histamine. And the reason we didn't like it is because it's not crazy, but we're not big on the immune hypothesis of Tourette syndrome. And since histamine is everywhere and obviously plays a role in things like allergy, et cetera, we weren't that excited about it. But as is often the case when you're what's called a positional cloner, which I am, is that you're taken to places that are surprising, and then you have to learn quickly about what's there. So we set out after finding this first to demonstrate that this wasn't a trivial change. And one of the ways to do that is that even though you presume this has to be non-functional, we can prove it biochemically. So we went to the lab and looked to see whether or not this molecule synthesized histamine, because it's the rate-limiting enzyme. And in fact, all these graphs show is that it does not synthesize. We lose the effect in the one bad copy. Now remember, they're going to have one good copy for mom and one bad copy, right? And so, and this is just showing that our biochemical approaches worked. But then we asked the question, so what's interesting about this enzyme is that the enzyme works in what's called a homodimer. So you have two copies that are made, and then they come together and they form the pocket that allows L-histidine then to be changed into histamine. And so sometimes when that happens, if the bad copy hangs around, then instead of just being loss of one copy, what you can get is the good copy of the protein binds with the bad copy, and you get worse than half. Right? And so worse than half is called a dominant negative mutation. So what this is showing, so if it was really half, we should get 50%. And what we found is that, in fact, if this was hanging around, I'll say a little bit more about that in a second, we got less than that. We got about 25% activity from the combination of the mutant and the wild type, suggesting this is dominant negative mutation. Now, we also started studying and found some interesting stuff, which suggested that even what's called haploinsufficiency, even the loss of one copy, would lead to decreased production of histamine, particularly in the brain. So what do we know about histamine? So we had to learn really, it's like, well, let's go see if there are any studies of histamine. And so we typed in the gene that we found, which was L-histidine decarboxylase, and a couple of articles came up, but nothing that interesting, except this is the rate-limiting enzyme, and this is what we can tell you about it. But if you go back and use study terms like histaminergic neurotransmission, neuropsychiatry, you get a huge volume of literature, and the volume of literature is all coming from the pharmaceutical industry. And the reason it's coming from the pharmaceutical industry is they've been thinking about targeting the system for the last 10 years. So it's it's highly expressed in brain, and then suddenly I thought, oh yeah, I've taken an antihistamine, and it made me really sleepy. This is a neurotransmitter, right? And the fact is it is a neurotransmitter, an important neurotransmitter. So it's involved in sleep-wake. It's involved in appetite regulation. And if you want to get the pharmaceutical industry interested in something, tell them sleep, wake, and wait. Right? These are huge. So they've been going after this for a while. And as they've been going after it and working on it, they have done, essentially, they've gotten us almost all the way around that circle in thinking about what would happen if you knock out this gene or if you have only one of two copies. So what they've done is they've created a mouse knockout of our gene. They have not only the knockout, which has no copies, they have the heterozygote, which has one of two copies, just like what would happen in our family. And they've done studies, behavioral studies, because they're doing neuropsychiatric stuff. So they've looked at things like what happens when you give dopamine agonists to mice that have decreased CNS production of histamine, and I'll show you an example of that in a second. So histamine receptors are not only in the brain, but they're highly expressed, particularly in regions that are relevant to Tourette syndrome. So I'm going to show you some expression data from our lab in a second, but very highly expressed in the striatum. So it's also clear that, so the way that the system works is that histamine is produced only in a small number of neurons that project from the hypothalamus, and this is our gene right there, L-histidine decarboxylase. It's the one thing that leads to the generation of histamine. It's then released into the synapse. There are a bunch of postsynaptic receptors, H1 and H2. And there's also a presynaptic autoreceptor called the H3 receptor. And what that does is when there's histamine that's released into the synapse, it will bind to the presynaptic autoreceptor and regulate histamine release. And this turns out to be the thing that's been targeted by drug companies. So every major drug company has an H3 receptor something, inverse agonist, so because they've been playing with histamine. Now, it also turns out that this H3 receptor is also on non-histaminergic neurons, and what it does is it regulates the activity, it modulates the activity of other neurotransmitters, including dopamine and serotonin and other things that are our favorite kind of molecules. It's where the lamppost is. But so this is a neuromodulatory process, and in fact what it has is it has the opposite effect. The release of histamine has the opposite effect of releasing dopamine. So when you release dopamine, what happens is that histamine is then released and it reduces the postsynaptic signaling involved in dopamine release. Okay? So now it also turned out, as I said, that there were already mice. So we're already now 3/4 of the way around the circle. We didn't have to make the mice, and they've done some experiments that we would like to do, although my collaborators at Yale wanted to make sure that the experiments that they did were repeated in a way that we could be really confident of. So I'm going to show you some of our data, some of which is just a replication of what the pharmaceutical industry has already done. So it turns out that mice with decreased histamine, and you can modulate this both using pharmacological agents that will block histamine release at the synapse. You can block H1 and H2, so you can recreate decreased histamine here, or you can block HDC, and they've done that because they knocked out HDC. And what was clear is that that rodents that have that, both mouse and rats, have increased sensitivity to dopamine agonists. So they have repetitive stereotype behaviors like gnawing, head bobbing, etc. So sometimes dopamine agonists will lead to hyperlocomotion, and in other times it will lead to repetitive behaviors that look an awful lot like human tics. And while face validity is kind of fun and interesting, there are several investigators that for many years have been looking at the relationship between these repetitive behaviors that are generated by dopamine activity and using those as a model for tics. So this is just data from Chris Pittenger, who's a collaborator at Yale, just making sure that all of this is just not sort of fluff from the pharmaceutical industry. This is a Western blot looking at protein, and what we find is that when we are looking for HDC activity, the activity of this molecule, it's really striking. So everyone said, oh, it's only in the hypothalamus, because the neurons are hypothalamic neurons and their cell bodies are in the hypothalamus and they extend everywhere to the brain. So you're only going to find HDC in hypothalamus, and what we find is a huge signal in the striatum. So for some reason, HDC is blazingly expressed, and not at the mRNA. I mean, this is protein level. So it's not just mRNA. There's protein in the striatum, and this is just a quantification of that, suggesting that there's really no difference between hypothalamus and striatum, but those are two regions of very high expression. So we went, and we had known that this was already done by a number of pharmaceutical companies, but we wanted to make sure, Chris, that we did it right. And again, so we looked at these knockout mice, and we asked the question, if you give them dopamine agonists, do they have repetitive stereotype behaviors? And the answer is yes. In this case, at these doses, the knockout, which is has 0 copies, had statistically significant increase in these repetitive behaviors, and not the heterozygote. But it turns out that as you up, we have some additional experiments that show that as you up the threshold of amphetamine, that even the heterozygotes will have increased sensitivity. And in fact, there are previous studies from the pharmaceutical industry showing that 1 copy of HDC in mouse brain will lead to a 50% reduction in histamine levels. And the reason that is that important, because one possible sort of flaw in this whole argument would be, even if you only have 1 copy of the protein, sometimes for enzymes the other copy will rev up and it'll give you just as much histamine as if you had 2 copies. So in this case, we know from the mouse, which has a very similar histidine decarboxylase model, that even just having 1 copy, this sort of excluding the notion of dominant negative, you only get about 50% histamine. So it also turns out that you can reverse this effect of these stereotype repetitive behaviors in rodent models, and you can do it. So if you give enough stimulant to any mouse or rat, you will get these stereotype repetitive behaviors, and then what you can do is ask, well, can I reduce those by using histamine as a neuromodulator? And this is a really simple way of doing it. I think, when was this? 1997. They just gave a bolus of the precursor to histamine generation in the gut, and it crosses the blood-brain barrier, and then it turns out that as they increase brain histamine, essentially these are the repetitive stereotype behaviors in the face of amphetamine, and this is what happens. Essentially, you just return to baseline levels by giving histamine back. So Chris was also really interested, I should have the name down there, Chris Pittenger again, was really interested in an endophenotype of Tourette syndrome called involving pre-pulse inhibition. So this is something that you see also in schizophrenia, but in Tourette syndrome, do you guys know about pre-pulse inhibition? So it's this neuropsychological construct and text where or test where if you make a sound, people will have a little bit of startle, but if you give them a sound a little bit before the big sound, you'll decrease the startle. So it's a quick and easy test to do, and they're well-defined ranges of normal. And it turns out that in family members as well as affected individuals with schizophrenia, you have impaired pre-pulse inhibition. And it also turns out that that's true in Tourette syndrome. Now, we went back to our original family, the odd family with 8 kids, and we did pre-pulse inhibition tests and found that, in fact, yes, that family has a significant decrease in pre-pulse inhibition with the HDC mutation. And then we did that in our HDC knockouts. And what you see is a dose-related effect that goes from essentially normal to the heterozygote state to the homozygote state with impairment in pre-pulse inhibition. So we're seeing it both in the adults, in the family members and in mice. All right, so where does this take us? So you might think, wow, that's quite a story off just a single genetic mutation. But in some ways, I think, so we have now a variety of activities going on at Yale sort of pursuing this question about what's the role of histamine in the generation or modulation of tics. And so we've done some PET studies in this family and also in the rodents, looking to see whether we can draw a link between the mutation itself as opposed to TS. And we're using some animal models to do more of an elaboration as we go around this cycle. But the most exciting thing to us potentially was that, so this is, in another context, if I stood up and said, okay, we found this one family, it's a mutation in histamine, I think we have very good evidence that that's doing something in this family, and it's a good brain-expressed molecule, and we see some stuff in mouse that looks an awful lot like what we're interested in, and we know we should be able to fix it by modulating something. So we're going to start a 10-year process developing pharmacological agents in order to test this hypothesis. You would say he's nuts. Because that would be nuts. And it would be premature to take all of those controversies. The reality, though, is that these medications that modulate histamine in a way that we would predict would make tics better based on this finding are already in phase 2 trials for disorders that are comorbid with Tourette. Right? So they're already being used for their quote-unquote pro-cognitive effects in trials for neuropsychiatric conditions that have essentially kind of the same morbidity as Tourette syndrome. So safety profile has already been established. They're already recruiting people for these clinical trials. They have compound. And so in this case, what we set out to do was to say, well, as long as you're doing that and you're looking at people who have, say, impaired attention, why not think about adding some kids who also have, not kids because we have to start with adults, but why don't we start with some adults who also have tics and Tourette syndrome? Because the costs, and I don't mean that just financially, but sort of the scientific costs of seeing whether or not this molecular pathway might have identified something that would work to moderate tics is very low and very fast. And so, in fact, surprisingly, several large pharmaceutical companies have been quite interested in moving ahead with this. So I hope that in a year I'll come back and tell you about the first clinical trial of an H3R inverse agonist in kids or adults. Actually, it will be adults at that point. And then we'll see whether or not this is right. Now, I have to say, as a child psychiatrist who went into genetics in order to be able to try to do something good for the kids that I see, just the idea that we could try this really has made my year. And this is something that, we found stuff for Tourette and autism before that still is in like really essentially right here after 6 years of work, where we've made almost no progress in really understanding anything that would lead you to think that you might have a treatment. And so in this case, the perfect storm has given us, I think, a really exceptional opportunity. But the final thing I'd say is that one of the reasons I wanted to give this talk is, we could be wrong. We often are. We're more wrong than right in the lab, and we just keep at it and figure even if we're right once or twice, that'll be enough. And so far, that seems to be holding up. But the reason that so we could really be wrong about this, and we for a whole variety of reasons. But what I wanted to point out is that this is sort of a paradigm, and hopefully a nice example of the fact that even if you're looking for rare mutations, or even if you're looking for common mutations, there's nothing restricted just to rare, is that the answer about so what to gene discovery, sometimes can be pretty interesting, right? So I think people are burned out. Genetics has been promising results for a really long period of time, and a lot of people, families in particular, don't want to hear about another study to find a gene. And I think a lot of physicians are as well. It's like, it does nothing for me in clinic, and I don't really care whether there's a 10% increase in risk for this polymorphism. And so, I'm here on a mission to say there's still some hunt left in the genetics dog, and whether it's rare or common variants, that thinking about genetics as an entree into mechanism, and from there into thinking in a novel way about treatment or intervention, I think that, we still have a ways to go and some real opportunities emerging in psychiatry. So that's the talk. But before I forget, I need to make sure not only to thank the folks that help fund this, but really it's that nothing in the lab is done without a tremendous amount of help from other folks. So Chris was leading the mouse studies, which has been really helpful. These guys, we do all of our gene discovery together as a group, which is really nice and fun at Yale. Maracanel and Rick Lifton. Hakan Arson gave us controls, which are really helpful, and these guys are in our sequencing core. These guys should really be at the top because they're the clinicians. And one thing about genetics is you can have this cast of brilliant characters over here, all my postdocs and students, and if we don't have patients in the lab, we have absolutely nothing to do. So Bob King runs our TS OCD clinic at Yale, made a huge contribution to this study, and Don Gilbert and Peter Hoekstra and Jay Tishfield, Gary Hyman are all folks that have contributed samples for us to test. And then these are all of the postdocs and stuff. And I just want to point out where Gulhan Erkan was really the lead on the paper that I showed you on L-histidine decarboxylase. If you're interested, it was 6 months ago, New England Journal, one of those short reports, and there's much more detail. We have a 60-page supplementary material. It's the most supplement per report I think ever done. One family, but it'll tell you pretty much everything about what we know about these guys if we don't have a chance to talk about it here. So thank you very much. I think we have a couple minutes. Is that right, for questions? Thanks.
That was so great. I only have 20 minutes for questions because it was so interesting.
Cool.
But I'll resist the urge. I've noticed before that antihistamines will induce restless leg syndrome.
Yes.
Folks, and how we treat restless legs if we want pharmaceutical helps get pramipexole.
Yes.
So that's an interesting connection here. And I was wondering, well, A, do you see that people with OCD or people with Tourette's, do their tics get worse with antihistamines? Does it happen in this family? And
Start there.
what neurons are expressing the histamine receptors? And I'll ask.
Okay. Yeah, I think I'm supposed to. I need to go to another website for 1 second. Sorry, and then I will answer that question because I guess we're doing a. Yeah. Is that? No, that's not it. That's not it. Oh, come on. There it is. Okay. My vulnerability has been blocked. That's good for a shrink. I'm not vulnerable. Okay, so so first question. So the question was, it was prefaced by an observation that antihistamines can induce restless leg syndrome, and there are treatments for that. And what do we know about antihistamines and Tourette? And the answer is that there's one other paper in the New England Journal about histamine and Tourette syndrome, and it's a case report from the mid-1980s about a small group of kids who had the onset of tics and Tourette syndrome with antihistamines. There are a couple of other case reports in the journals Neurology and in Movement Disorders of kids who got, and adults who had histamine who either developed dystonia or tics. So now that seemed good to us. Now the flip side is that anytime you start thinking about just up or down, you're going to be in trouble. And so it actually turns out that we know clinically that some kids don't do well at all during allergy season. We give them an antihistamine. It may be the allergies, it may be the antihistamine, they have terrible tics. But there are other kids where you give them antihistamines and they have tics and it quiets things down and they seem to be fine.
How about
Yes.
this family, though?
In this family, so we didn't give them antihistamines. We are hoping that we might be able, that they will be interested in coming back if we can work with the pharmaceutical companies who might be providing us compound to see what happens. This family, you know, I think, so one of the issues is that they already have decreased histamine and they've lived with it for, in their CNS for a very long time. So I'm not sure what it would mean to further block this, but we certainly will give it a try. And what we'd like to do, we have some baseline PET studies, but we'd like to look at their response to dopamine agonists. Yes?
So, that was a fabulous talk.
Oh, thank you so much.
So, have you screened, I'm sure,
Yes.
this is in the article, but have you screened other
Yes.
large samples of replication? You mentioned that this is rare.
Yes.
So have you found this mutation or in other exons of this gene?
Okay.
in
Yes.
Either TS or OCD or, the spectrum of something?
Yes. So what we did initially was we asked in whether or not this mutation, how rare was this mutation? And so we looked for this specific mutation in about 3,000 other people. About half had Tourette and about half did not. And we didn't find it anywhere, which doesn't, rare mutations are rare, whether they're related to disease or not. So lots of people, everyone here has private mutations. So that doesn't really mean a whole lot. So then we asked the next question, do we see a lot of variation in this gene? And so we've sequenced it so far in about 700 folks with TS. And we found a couple of missense mutations that we're modeling now to see whether it has an impact on, because now we have all the assays set up and we can look at it. But there wasn't anything blazing. We found no other stop codons. Now, so conversely, we had sort of ongoing a large-scale, relatively large-scale CNV study of Tourette, and by another lab member that essentially nearly simultaneously. So we had a whole bunch of rare CNVs in Tourette cases and in controls. And like many CNV studies, copy number variation studies, we weren't seeing a lot of difference between cases and controls, sort of writ large. So we did pathway analysis to ask the question whether or not these were organizing themselves into any known pathways. And depending upon the tool you use, you get slightly different answers. But in each group, with each tool we used GeneGo, we used the David program. Among the top three were histaminergic genes. So we have multiple rare CNVs that are not present in siblings, that are present in genes that are involved in either histamine generation, histamine signaling, or histamine metabolism. So that's where we're headed now, is to see whether or not that accumulated group are affecting function.
So does this family have any unique CNVs?
No.
And is the location close to the schizophrenia 15q?
No.
It's not.
No.
It doesn't matter.
And there's absolutely nothing else in this interval that we can tell. So after we did the exons, now that we can do new stuff, as we went back, we pulled down the entire interval, sequenced it, and there's just nothing there. This is what's segregating with this family. Now, the maximum possible log score is only 2.1, So there might be some other region of the genome we could be completely barking up the wrong tree, but in this region, this is what's segregating.
2.1 in this family.
Yes.
Pretty good.
Yes.
Morphologic or neurodevelopmental abnormalities associated with the HDC knockout?
With which?
For the HDC knockout, or just believed to be a purely functional phenotype?
Right now, so the most of what has been done in the pharmaceutical industry was interested in behavioral effects. So there's not a lot done on morphology. There's some work that's been done on assaying how much histamine is in the CNS, but there were no gross morphological abnormalities. But we won't settle for that. We're going back and we have the mouse, and we're looking very closely. It's not gross, but we have questions about what's going on, particularly in the striatum, that just people had not really focused on previously.
Which cells are making it in the striatum?
There's no cells making it in the striatum.
It's all from hypothalamus?
It's all from hypothalamus. And the question about why there's so much HDC in the striatum, we have no idea.
Well, I mean, there's the, if you look at the hypocretin system, it's not they eat, it's they get up and eat, wake up and eat. Peptide or histamine, maybe do something similar.
Yeah.
Wake up and start looking for food.
Which is what I need to do. Yes?
Are the receptors located on both the indirect and direct pathway neurons in the striatum? So that it's coming in.
Yes, so what's interesting is that they co-localize histamine H1 and H2 co-localize with D1 and D2 receptors postsynaptically. And they really they have countervailing properties. It's been best worked out for D2 receptors, but they shut down signaling from D2 receptors when they're stimulated, and they appear to do the same thing in the direct. So you can't really, so at first we thought, oh, well, maybe it's an imbalance in the pathway, like, it's like but in this case, but it turns out it's not really possible. I mean, the only thing that we can say is that this system is very closely related to regulation of dopamine in the striatum. And so one of the reasons that we were so interested in the possibility of this working pharmacologically is we already know that if we block dopamine directly, we can help tics. So even, the notion that if we can modulate it indirectly by manipulating histaminergic neurotransmission, we might get a similar benefit without, we hope, the disadvantage of direct blockade of dopamine receptors. Yes.
You're talking about dopamine blockade with antipsychotics. Because the drugs are so dirty,
Yes.
Are you finding or thinking about sort of different atypicals because they have more histaminergic activity, are themselves?
That's an interesting question.
More or less?
So the question is, I'm sorry, I keep forgetting to repeat the question. The question is, since atypicals are so dirty, do we think that maybe some aspect of the efficacy of the antipsychotics has to do with the histaminergic properties? Yes? And the answer is no, we haven't been thinking. But I mean, it's a good thought. We just haven't been thinking about it. Any other questions? Well, thank you all for your attention. I'm sorry I kept you a few minutes late. Appreciate it.